Good morning, class. Let's go ahead and get started for the day. So for today's lecture, we're going to be focused on talking about tools to defeat the coronavirus. So hopefully this one will be a relevant lecture that you guys are able to take with you after this class. So for today's outline, I'm going to talk about the history of vaccines and how they were developed. I'm going to talk about some of the main vaccines that have existed before, but how our technology has changed and how we're able to basically use some of the new technology to fight against COVID. I'm going to talk about monoclonal antibody therapies and how that can be used to treat people that are sick. And then I'm going to talk about COVID variants and what this means for COVID therapies. So for the learning objectives, know the different kinds of vaccines that exist and how they work. Most of their mechanisms are very basic. And now that you guys have an understanding of the central dogma of central biology and basics of immunology, you should have an understanding on how these work. And if you don't, ask me. I want you to understand the differences between a primary and secondary immune response, because this is really important when it comes to understanding why vaccines are so effective. I want you to understand the basic biological principles of monoclonal antibody therapy and how some of these antibodies are derived. Understand what COVID variants are and what this means for COVID vaccines. And then just to reiterate some stuff that I've talked about in the past, I'm not going to talk about antivirals or anti-inflammatory drugs in this lecture, but I've covered them in previous lectures when they were relevant. So please remember how those work, because you will end up getting test questions on those. So to talk about viruses or vaccines, we need to talk about smallpox. So smallpox, as seen here, as the virus itself, and then when it manifested in disease in humans, is caused by the variola virus. And it's thought that it originated about 10,000 years ago in Africa. And studies have shown that back in the day, when people would end up getting infected for about 2 weeks, about 30% of those would end up succumbing to the infection. As the old world became connected through merchants and the new world by European conquistadors, smallpox was able to spread throughout the world, and it was, found through all regions in the world. It's estimated that over, the 10,000 years that it's been around in the human population, that it's killed billions of us. But just in the 1900s alone, it's estimated that 300 to 500 million people were killed. So the origins of vaccines, really, at least the concept behind, trying to form immunity, began in China in 1022 AD. So before the invention of modern medicine, this method of vaccination was called variolation. And there was two forms that were done. So the first form that was invented by the Chinese would blow dried smallpox pus into the nose of patients. And the second form of variolation was when doctors would actually pop some of these pus lesions, which had active virus in them, and then they would take just a small bit of that and then plant it under the skin of an uninfected individual. And the whole point of both of these resulted in relatively mild infection in these individuals, but it was for the most part survivable. As I mentioned, when people were unvaccinated or unvirulated, 30% of the population would succumb to the infection. With this new type of way of educating your immune system, 3% of the people would end up dying from the exposure, which, it's still something, but it's not as bad as it was. Then it wasn't until Edward Jenner came along, who was an English physician, that basically is credited for inventing what we now know as vaccines or vaccination. So when he was training to become a doctor, he noticed that some of the milk maidens, when they were milking the infected cows that had cowpox, were getting cowpox all over their hands, but He also noticed that they never developed smallpox. And so just to, mention this, because I haven't covered any of this in any of the previous lectures, smallpox and cowpox, they come from the same family, but cowpox is really, meant to infect the cow only and create severe disease, whereas smallpox is really infecting humans and creates severe disease. But cowpox can infect humans, but we don't succumb to the infection, or we don't die from it. And so he had this idea of taking pus from the cow lesions that were on the infected milkmaiden's hands and doing what previous doctors had done in the past, where they would plant some of that weakened virus under the skin of the uninfected individual. And what he ended up noticing was that people that he ended up treating with this cowpox pustules never developed the smallpox virus. And because he was using the cowpox virus instead of the smallpox virus, instead of it being called viriolation, it was called vaccination. And so when he did all these tests, basically, after, really treating people with this new method and then exposing them to the smallpox and seeing that they didn't end up developing any kind of infection, or if they did, it was very mild and their body was able to clear it. It was then that, vaccination really developed and the government started to get on board. What was the logic behind putting the pathogen into the patient as a form of vaccine? So the whole point of it is so people were dying from it back in the day. They didn't, really know anything. Viruses weren't even known to cause disease at this point. It wasn't until really like the last century or two that people were able to do this. It was so that people would not die. That's why there was this form of vaccination that was done back in the day. So here I have a timeline of vaccine development. So, Edward Jenner is, credited for the first vaccine against smallpox in 1796. And then, as you go through time, it's not until, almost 50 years later that the next virus or vaccine is developed. And this is developed in the 1950s by the humanitarian and scientist Jonas Salk. And he's a great guy because basically when he invented his vaccine, he didn't patent it and made it basically free so anybody could use it, so they could eradicate the disease. As we continue to go through time, we see that about 5 years later, the measles vaccine is invented. And then about 10 years after that, then we have the rubella vaccine. And these are all vaccines that we routinely get when we're babies, so we can go ahead and have immunity against these particular diseases. And then again, we don't have another vaccine until 2006 with HPV. And I've highlighted this in a previous lecture where basically this particular vaccine has 90% efficacy when it comes to preventing any kind of outbreak or infection. And then I forget when the influenza vaccines were started, but there was mass production in 2009 for the swine flu outbreak that was occurring during that time. And so basically with the invention of all these types of vaccination, especially within this last 100 years, the world basically got together to try to eradicate some of these diseases. And the first one that the World Health Organization wanted to target was smallpox. And so here we have a schematic of the status of smallpox in 1959. And so we can see that the majority of countries that are plagued with smallpox are mainly in third world countries, but then you also have a couple that are in Europe. And so the world decided to get together to eradicate smallpox. And so the countries that are highlighted in red basically contributed money to go ahead and fund the program so they can go ahead and really try to get this campaign going and just get rid of this altogether. And so it was a very collaborative, program to try to get rid of this disease. And so efforts began in 1967 and lasted for about 10 years. And so it was a very laborious because basically, again, all these people were in third world countries. So people had to go and travel to these places, and basically go house to house, market to market, places where people would gather to basically, promote or tell people that they should get vaccinated, and it's free. And so to contain the virus or any kinds of outbreaks, if they were aware of somebody that was infected, they would usually keep these people in isolation. And then if they knew that this person was in close contact to, other people in their family, they would try to vaccinate everybody within their household so they didn't develop it or a new outbreak would begin. And then on top of that, if there was a people that were in a particular community that it was known that smallpox existed, then they would go ahead and vaccinate the neighboring 30 households. And this is, to really prevent the spread and keep a whole community immune against the virus. And so this took a long time, but it wasn't until 1980 that the World Health Organization basically announced that smallpox is officially cleared from the human population. And so currently the virus only lives in two places. It lives in the CDC, Center for Disease Control in Atlanta, and a lab in Siberia. And the whole point of really keeping these around is in the event of, some kind of outbreak, if somehow one of these labs were compromised and terrorists were able to get a hold of some of these and, unleash a, bioweapon, these labs would be able to manufacture diseases quite quickly because they already have the particular strain present. So after the eradication of smallpox, the World Health Organization wanted to focus its efforts on eradicating polio. And basically, this is one of the viruses that causes severe neurological deficits because it likes to infect neurons specifically in the spine, and the spine, plays a role in motor function, amongst other things. So the program began in 1988, and so we see where a lot of these countries never had eliminated polio, where it was quite present within the population. And then here I have a diagram of what the countries that have never eliminated polio in 2014. So basically, most of the world has been cleared from polio. It's only a handful of countries that have not cleared it. And, there's some cultural reasons that play a role in this. And when I talk about anti-vaxxers in a different lecture, I'll go ahead and highlight some of those reasons or rationales. So for conventional vaccines, in an ideal world, we would want a vaccine that would be safe for everybody, so it wouldn't cause any kind of harm or illness or disease in the patient. And we'd want it to not only be protective against being infected, but having sustained protection where you're able to have immunity against it for life. And this is all mediated through your immune system, your B cells that are able to produce neutralizing antibodies, and your T cells that basically are able to recognize infected cells and kill them. And so an effective vaccine is going to be able to mediate protection through these immune mechanisms. And so we haven't really changed, what kinds of vaccines we use for the last 60 years until really now that, COVID has hit, and we needed to basically change our strategy when it comes to dealing with viruses and educating our immune system. And so basically the main approaches that we typically had prior to COVID were using a live attenuated or weakened virus, or by using a killed virus or viral proteins from that killed virus, and injected with something called an adjuvant, which I'll discuss in the next slide or 2 slides. So to highlight each one of these particular vaccines, because they're very important, they're still used today, but even though, efficacy is still a problem with a couple of them. So again, a live attenuated virus vaccine has a live virus in it, but it's been weakened in some way. Usually it's through some kind of chemical modification. And so if you have some kind of chemical modification of the virus, or you got it from a weakened source, just like the classic example that I talked about with Dr. Jenner and the cowpox, then you're able to infect the patient. Because it is active infection, the virus is able to replicate, but because it's weaker, your immune system should be able to readily clear it. And because you mount that whole response where you have the innate recognizing it and then educating the adaptive, you're able to really create a strong memory response. And so with a lot of these virus or vaccines that are used for some of these viruses, such as the ones I have listed here, you typically only need 1 dose of them, or you might need a booster like 5 or 10 years later, typically. But the main risk for these kinds of vaccines is because, again, it is still a live virus. Even though it's weak, there's still that potential of it causing severe disease. And usually the people that are most susceptible to this kind of disease are people that are immunocompromised. And that could be for, because they are infected with HIV, they're on some kind of immunosuppressive therapy, or they're a cancer patient that are on chemotherapy. So it's good and bad when it comes to these particular types of vaccines. The other type of commonly used vaccine before this COVID hit are these inactivated or subunit vaccines. And so for these, they take an intact viral particle, and it's living, it's infectious, and then they'll end up treating it with some kind of chemical or radiation to basically destroy the genetic material. And as we remember from the viral lectures, in order for a virus to replicate, it not only needs to get into the cell, but it needs to deposit its viral genome so it can go and hijack the host machinery. And so if it doesn't have viral genome present or functional, then the virus is rendered inactive. And so this is what an inactive vaccine would look like. It's basically a viral particle that its genetic material is destroyed. Some subunit vaccines will actually not only go take this inactive virus, but they'll go a step further and actually grind it up and chop it up into pieces. And so that's what a subunit vaccine is. And so either one of these will end up being injected into a person with this thing called an adjuvant. And an adjuvant is for stimulating the immune response. So scientists don't really understand this, but for whatever reason, when you end up introducing just an inactivated virus or the subunits of the virus, it doesn't really create a strong immune response. And it might be because you have phagocytic cells like macrophages coming in and just quickly eating it up. So you need to stimulate the innate immune system. And what scientists have found over the years, and I think they found this back in like the 60s or 70s, that they add these adjuvants to basically stimulate those pathogen recognition receptors that are going to mount that immune response. And so the only adjuvant that's currently approved in vaccines is aluminum salts. And so here I have a list of examples of vaccines that basically utilize this kind of mechanism for introducing the virus to, or viral particles to, the immune system, so it can go ahead and form some kind of immunity. The major problem with this, though, is it's a very poor immune response to these kinds of viruses. And again, you need some kind of adjuvant to really stimulate this. And because this type of vaccine is very poor at stimulating the immune response, sometimes you need multiple rounds of administration of the vaccine. All right, so that was conventional vaccine. This is what we've been using for the last like 50 years or so. So how do COVID vaccines work? So it's really dependent on, which vaccine you're talking about, because there are so many out there on the market. And so here I have the main players that are involved in vaccine manufacture and distribution. And so we're going to talk about the mRNA vaccine, which I'm assuming most of you guys have already gotten, because that's the one that, our government's paying for, so we can go ahead and get it. You have adenoviral vectors, and then you have recombinant protein and adjuvant mechanism, just like the ones that I had described previously, but they've changed it up a little bit. So how does the mRNA vaccine work? And so this is really important because, again, you guys are going to be the source of knowledge for some of your family members. And it's a good idea to really understand what you're putting into your body. And I know that there's lots of fear when it comes to some of these new technologies. And so I'm hoping that by explaining how some of this work, and as well as me giving you the previous background with the previous lectures on understanding some of this terminology and how this works, that you'll be able to help alleviate some of that fear in either your families or friends, or even yourself. So here I have a nice schematic of how the mRNA vaccine works. And so I'm going to emphasize this over and over. The vaccines that are on the market right now target the spike protein. Again, the spike protein is the main protein that facilitates entry into a host cell. And so studies have shown that when it comes to the immune response recognizing the different parts of the virus, the one that the immune system is able to respond to the best are the spike proteins. And so that's why that's the main target. And so with these mRNA vaccines, they basically have different sequences that encode for different parts of the spike protein. And then they're going to be placed within these lipid nanoparticles. And when you give this into a shot, these nanoparticles are going to be able to bind to the host cell, deposit the mRNA into the cell, and the host cell machinery is going to be able to basically recognize this mRNA and create viral proteins. These viral proteins are going to be expressed on their cell surface through the MHC complex, which I talked about with adaptive immunity. And so that is how your immune system is really going to kick off being able to recognize something foreign in the body and start to develop some of that immunity. And so I want to highlight, those two. So that was a quick summary. So I want to highlight a couple of key parts into, how this actually works, because I feel like, at least from my family, I've gotten a lot of questions like, how does the mRNA vaccine work? How does it get in? What is it doing? And so hopefully by me reiterating some of these points, it'll solidify in your mind how this works. So basically, the mRNA, so RNA in general, and I've emphasized this multiple times, is very unstable. It will readily get degraded. And so it basically needs to be protected in some way. And so the way that companies have designed this particular vaccine is by surrounding the mRNA in these with a lipid coat. And if you remember from a couple of weeks ago, when we were talking about the structure of the cell membrane is also composed of lipids. And so likes dissolve in likes. And so when these lipid coats end up getting close to the lipid bilayer of the host cell, it's actually able to bind to it because it thinks it's also part of the lipid or part of the lipid membrane. And so just like when a virus ends up binding to the cell surface, it basically will fuse with the host cell membrane and then deposit its genetic material into the cytoplasm of the host cell. And then the host cell machinery is going to convert that into protein. And so this particular mRNA vaccine encodes for many different sequences of mRNA. So the spike protein itself is composed between 300 to 500 amino acids. And I can't remember if I emphasized this in the adaptive immunity lecture, but when a T cell and a B cell are recognizing antigen, their receptors only combine to like anywhere from like 8 to 20 amino acids. And so instead of just having one mRNA within these protein coats, they have many sequences that are going to encode for different parts of the spike protein. And so, going back to the central dogma, this mRNA is going to end up getting translated by the ribosome and converted into protein. And because it's only a part of the virus, it's not the whole virus, it's not going to make you sick. It's not going to be infectious. There is no virus that's going to get made. It's just a part of the virus, and it's the spike protein that's being made. And it's going to be different parts of that spike protein that are going to get made. And then, going back to the MHC diagram that I had in the adaptive immunity lecture, basically the body's going to recognize that something is not right within the body. And so these proteins are going to get loaded onto these MHC 1 and 2 complexes onto the cell surface. So your adaptive immune cells like T cells can come and recognize that foreign substance. Because it's going to recognize that foreign substance, it's going to end up mounting an immune response. And so that's why there are some people that have symptoms after getting some of these vaccines. And symptoms aren't a bad thing. People, complain, oh, I don't want to take the vaccine because it's going to make me sick. If you're getting sick, it means that your immune system's working, and that's a good thing, because that means you're going to end up generating a memory response against the virus. You're educating your immune system so it can actually see the virus when it actually encounters it. I have a question. So we're just injecting mRNA with instructions to make spike proteins so that those are recognized. Exactly. Yes, exactly. So that's how we're, educating our T and B cells. So really understand this process, because, most people, at least in the United States, are going to get this kind of vaccine. So let's talk about the other type of vaccine. One more question. Is it a small portion of the RNA, right? Not the whole thing? Yeah, so it's a small portion. So the mRNA is going to be a small portion that encodes for part of the spike protein. So the spike proteins, I just said about 500 amino acids big. So instead of, a vaccine that's going to encode for all 500 amino acids, it's only going to encode for some of it. And so when it gets translated, only a piece of that protein is going to end up getting synthesized from that mRNA. So for the adenovirus vaccine, so these utilize a viral vector. So yes, you are being injected with virus with this particular vaccine, but this virus is harmless. It's not the way that scientists have structured it is it basically will infect a cell and only infect once. It's not going to be able to replicate. And so here in this diagram, we have, the typical structure of COVID. And so like I'm going to emphasize over and over again, the whole point of this vaccine is to educate your immune system to the spike proteins. And so here is highlighted that spike protein within the coronavirus genome. And so scientists will basically take that section of it and then put it into the adenovirus. And so once you inject this virus into the body, the adenovirus is going to be able to bind to its respective cell. And remember, all viruses need some kind of receptors to end up getting into a cell. So I can't remember what the adenovirus receptors are, but besides the point. It binds to the receptors and then deposits its genetic cargo into the cell. And again, the way that this is designed, the virus genome that's being deposited in here does not have instructions to actually assemble this virus. All it has is the instructions to tell the host cell to make spike proteins, just like the mRNA vaccine. It's just a different way of being able to get this mRNA sequence into the cell so it can be translated into a protein. And so once the spike proteins are translated, just like the previous slide, as so, the viral proteins are going to end up being loaded onto the MHC, so the T cells can come in and recognize this, and then they're going to go ahead and basically start to educate the rest of the immune system on what to do. And so T cells, again, talk to B cells so they can go ahead and generate antibodies. And so these antibodies are basically going to be specific for the spike protein. So if any of the antibodies ever encounter the virus within the blood, they'll be able to readily bind to it and neutralize it. Somebody asked, and the spike protein is the first thing that is seen on the virus. So it's a very easy part of the virus to be recognized by. Exactly. So our immune system mainly recognizes, the adaptive immune system mainly recognizes the structural proteins outside of, most viruses in general. But for coronavirus in particular, it sees mainly the spike protein. I think it can recognize the envelope membrane as well, but the majority of the sequences that the T cell and B cell receptors recognize are the spike proteins. And they're the ones that elicit a stronger immune response, known as immunodominant epitopes, but you don't need to know that. All right, so that's how adenovirus vaccines work. And so the last kind are going to be these recombinant protein vaccines. And so these are going to be similar to how I described the conventional vaccines, except some of these companies are kind of taking a different approach. Instead of heat inactivating and chopping up the viral particles and injecting it into the cell, they kind of went a different approach. And so here what they ended up doing was basically pooling a bunch of spike proteins together, anchored through these lipid nanoparticles. So basically the nanoparticles in the center, and it kind of just holds all together the different spike proteins that are made. And so the way that they do this, which I found interesting, is that they genetically modify an insect virus. And then they basically, are able to create a lot of virus from this insect, because all viruses need a host cell to infect, and so they're going to be infecting an insect to do this. And so once they have enough of virus that's made, then they basically fragment it and then purify the spike proteins from it. And so they administer these spike proteins into the blood, and then antibodies, or the body's going to recognize a foreign substance. So instead of it, being taken up by a cell, it's going to recognize that there's something foreign floating in the blood. And then usually your innate immune cell will recognize that. And then, educate your T and B cells, and then your B cells will start to generate antibody against them. So how effective are COVID vaccines? So, it depends on the type of vaccine that's being administered and the company that's doing it, as well as the technology. And so if you look at the different, vaccines that are on the market, the ones that seem to be most effective are the mRNA vaccines. And it's a very novel way of being able to try to educate your immune system. We've had this kind of technology for decades at this point. I've even used it in the lab many times before, where you're just trying to introduce some kind of substance into a cell. But now we're actually seeing this translation from the bench that scientists are doing to humans, which is, I find, amazing. And this is one of the reasons why we're able to develop a vaccine so quickly, because the technology has already been present. And then with the invention of being able to sequence things so fast now, being able to sequence the viral genome and trying to understand some of those sequences that are really important for the immune system to recognize, we're able to do this and put it all together and, create something so beautiful that's really, helping to educate our immune system so we have less death in the world. So the mRNA is the most effective by 95, 94% respectively of these two, Pfizer and Moderna. This is going to be followed by the adenoviral vectors that I just gone over. And then the ones that are the least effective, but still relatively good, are the recombinant protein vaccines. And so I want to put this in perspective, because even though you see there's a decrease in the type of efficacy of these kinds of viruses, we get the flu vaccine yearly, but most of the time the flu vaccines aren't all that effective. The flu virus itself is very error prone when it's replicating, and so it's constantly mutating, and so it's very hard to educate your immune system for something that might not actually be there anymore. And then on top of that, the proteins that the immune system recognizes with influenza is the area that is always changing, because there's two parts. There's a conserved region to the virus, and then there's a non-conserved part. And our immune system recognizes that non-conserved part when it's trying to fight against the virus. And so if you have a viral particle that's constantly mutating and changing the proteins that's there, then your immune system can't recognize it as well. So typically the efficacy of these vaccines are 40 to 50% on a good year. Most of the time it's a bit lower. I think the best I've ever seen it was around 60%, but that doesn't even come close to most of the vaccines that we currently have on the market. So would it mean that mRNA vaccine is not a super? Yeah, right. Thanks. Yeah, I highlighted that, but I can emphasize it again. This mRNA vaccine technology is not a new thing. We've had this for some time, and that's why we're able to, readily do this. All right. So what does this actually mean, though, in the context of these new variants that are coming out when administering this vaccine to people? Somebody asked me before I get into this, can I define viral protein? Yeah, so viral proteins are basically any proteins that make up the structure of the virus. So if we go back to the intro to virology and the coronavirus lecture, the ones that are going to be most recognized are the structural proteins. So, the spike, the envelope, the membrane, and the nucleocapsid protein. So those are the viral proteins. Those are the ones that are not found in humans, and that's what the body is going to recognize as foreign. So, we're still learning with all of these changes that are occurring while the virus is replicating. We still don't know what this means for vaccines. A lot of studies are still being performed to answer these very questions. But when you really think about how the vaccines are working, what is really going to determine the efficacy of these vaccines is where the mutation is in the virus. And so, again, the vaccines that we have developed are against the spike proteins. And so if you have a lot of mutations that are within the spike protein, then it's going to make the vaccine a lot weaker when it comes to educating it, because again, the virus or the immune system recognizes very specific sequences. of the amino acids that are within the spike protein. And so if you have amino acids that are drastically different, or there's a lot of amino acids that are changed within the actual spike protein, then it's going to be harder for your immune system to recognize it. So it's going to make the vaccine less effective. With that being said, if the mutation is of a similar amino acid, so if we go back to when I was talking about the different amino acids that exist, if the structure of it is relatively similar to what it previously was, the immune system will still be able to recognize it. So if the two amino acids have the same charge, or their structure is the same, or the polarity is the same, then the efficacy of the vaccine really isn't going to change all that much. But if those structures are drastically different, then it will change the efficacy. But again, a lot of studies still need to be performed when it comes to trying to figure out, what this is. I did pull up just an example of data that are where people are currently trying to test the efficacy of the virus, of the vaccines to the new strains of the coronaviruses that are coming out. But before I get into that, it's important to note that the CDC and World Health Organization are trying to change the names of these new variants that are coming out based on the Greek alphabet instead of being by region, because, with all the ignorance and racism we have in the world, people are going to start associating specific viruses with specific regions and specific people, and we don't want that. So now they're going to be changing it more to the Greek alphabet. So the one that you may have heard recently in the news is the Delta variant. And so Pfizer and AstraZeneca are both have both tested their efficacy of the particular vaccines against this particular strain. And so we see that for both of these, after the first dose, there's relatively low efficacy when it comes to trying to recognize this. However, once you get that second dose, which you need to get to really boost that immune response, the efficacy increases to 60 and 88% respectively. And if we compare this to the efficacy of what I had shown before, I believe it's about 95% with the, or 94, 95 for the Pfizer vaccine. So it does decrease a bit. And again, it has to do with where that mutation is within the virus. Let's see, I got some more questions. What does a new strain necessarily mean? Do they still have the same spike proteins, or immune systems are educated on? So they have, so the proteins themselves are still considered spike proteins because there's a gene that encodes for the spike proteins, but the amino acids that make up those spike proteins are different. And so that's where you get these different variants. So the first virus that came out had, we'll give an example, say like in one of the regions had an amino acid called valine. And with the different rounds of replication that occurred, sometimes point mutations will occur within the virus, and say that valine is now a glutamine or a aspartic acid or something like that. And so that change is still, part of the spike protein, but the amino acid that makes up the spike protein is different. And again, the T and B cells only recognize very specific sequences of amino acids. And so if those amino acids are different, then the immune system is going to have a harder time at recognizing that. Do you think more research will be done of the efficacy of the vaccine for new Lambda strains? I'm sure with all of these, there's going to be more research done to test the efficacy of all these different vaccines. But I think the money is going to go, or research is going to be done more towards the ones that cause more severe disease. Yeah, but we'll see. Time will tell. And so what we really want to, what I want to emphasize is this concept of primary versus secondary immune response. So I've shown you guys this diagram before, and I'll quickly walk you guys through it again. So if your body has never seen the pathogen before, so we'll take the coronavirus, for example, you'll have the initial exposure to the virus. And then with respect to time, because the immune system hasn't recognized it yet, the virus is going to start replicating. And then it's not until there's a certain level of virus that's in the body that your innate immune system is going to start kicking in. And so this is going to be the first stage of the infection actually establishing itself in the host. And then, once your innate immune system recognizes it, then it's going to start educating your adaptive immune system. And this takes about 5 days. So there's that 5-day delay before your adaptive immune system actually starts to get educated. So that's 5 days of the virus being able to replicate in your body. And so then once enough of your B and T cells have been educated, then the adaptive immune response comes and starts to clear off the infection. And then once the infection is cleared, the B and T cells will undergo something called clonal deletion, where the majority of them will end up undergoing apoptosis, but a small fraction of them, about 10%, will end up living, and those are going to be your memory cells. And these are going to be your important cells that are going to be able to recognize a secondary infection. And so, here, this part of the graph is analogous to this part of the graph. But what I want to emphasize before I talk about secondary exposure are the vaccines. And so instead of this whole process of the innate educating the adaptive, in the process of you getting very sick because your body's trying to fight against a pathogen, the vaccines basically skip some of this. There's still that antigen presentation that takes place where the spike proteins are going to educate the T and B cells, but during that process, you're going to experience either very mild symptoms or no symptoms at all. And so that's one of the beauties of getting vaccinated is you can educate your immune system without actually getting sick. Versus, the people that haven't gotten vaccinated and then end up getting this primary infection. And if they're one of the high risk people, then, they're not going to be able to come back from this because there's that 5-day delay of the virus being able to replicate, and your adaptive immune system coming in and trying to clear it. And so with either people that have recovered or people that have been vaccinated, once they actually see the virus for a second time or see a component of the virus, such as the spike proteins through vaccination, then what's going to end up happening is your B and T cells are going to be able to recognize it and basically sound the alarms a lot quicker. So instead of this, innate immune system talking to the adaptive immune system 5 days later, you already have these memory cells that have been formed that are going to be able to recognize it. And so once it recognizes that there's this foreign substance that it's already seen in the past, it's going to be able to mount a response not only quicker, but also stronger. And so this is why vaccines or primary infections are able to get cleared so quickly. It's because you already have these memory cells present. And so again, when they see that pathogen again, then they go ahead and mount a very strong and fast response. And so that's why people that get vaccinated, yes, you can get infected even though you're vaccinated, but because you already have these educated memory cells, they're able to clear the virus so quickly that you're barely going to experience any symptoms at all. And if you barely experience any symptoms, that means it's going to be harder for you to actually spread the virus to people. So the last type of therapy that I want to talk about are these monoclonal antibody therapies. And so just to highlight, antibodies again, the whole point of antibodies is to basically neutralize or prevent viruses or any pathogen really to bind to and get into a host cell so it can replicate. And so here I have a diagram of the coronavirus that has IgG bound to it. And it's recognizing specifically the spike proteins that are on the virus. And so if you have all of these antibodies recognizing these spike proteins, then it's the spike proteins can't bind to the ACE2 receptors and thus cannot get into the cell. Someone asks, in other words, the virus will show up in your body, but it will get whooped. Yes, yes. That's the whole point of vaccines, or at least the effective ones. But now, with enough people getting infected, doctors can now infuse infected patients with antibodies. So, they typically do this for people that are part of that high risk category. So what they end up doing is infusing these patients with these things called the monoclonal antibodies. And so as the name suggests, by mono as one, it's going to recognize one specific antigen. And the antigen is typically going to be the spike protein, because you want this process to happen to prevent the antibodies or the virus from being able to enter a host cell. And so basically the antibodies that are infused into infected patients come from three main sources. So you have people that have been infected before and have recovered and basically donate their plasma. And, going back to yesterday's lecture, your antibodies are found in your plasma. And so, some donors will end up just donating their plasma, and then that plasma can be put into infected patients so this process can happen. We're able to also generate these monoclonal antibodies by these humanized mice. So basically, scientists are able to genetically modify mice where they're producing antibodies that have human structures. Because they have the human structure, when you put it into the human body, your body's not going to think it's foreign because it's going to think it's self. And so you can basically challenge these mice with a specific antigen. Typically, it's going to be a spike protein, and then they're going to end up generating an immune response against it. And then once the mouse has recovered, they're going to be able to have a lot of circulating IgG specific for the spike protein in the blood. And then scientists are able to basically purify that antibody out, and then doctors can go ahead and infuse that into patients. And then the last kind of way that or source that these antibodies are derived from are these things called hybridomas. And so hybridomas are a type of cancer B cell lines. Remember, B cells are the one that produce your antibodies, but you're able to make these lines that are able to create antibody against a specific antigen. And so again, the specific antigen is going to be the spike proteins. And so you basically fuse these B cell lines that are able to produce your antibody against whatever antigen you want, fuse it with the cancer cell line. So there's basically two stuck together, and then they secrete a shit ton of this antibody. And then scientists are able to basically purify that antibody from sorry, I didn't say this. This is all happening in a petri dish. It's called in vitro, the fancy words, but all of this is taking place where your cells are growing on a dish. And then they're just secreting antibody into the solution that the cells are living in, and that solution is taken, and then the antibodies are purified from everything. And so all three of these can be used to be purified and infused into patients that are infected. So you basically can not only neutralize the virus by the antibodies binding to it and preventing the ACE2 receptor spike interaction, but also by having your innate immune cells come in and eat the virus. So I've emphasized this a couple of different times. Innate immune cells have these FC receptors. And so what ends up happening is when you have your infused antibodies that are specific to the spike proteins, they're going to end up binding to the spike protein. They're going to end up surrounding the whole virus as so to prevent it from being able to get into the host cell. But these circulating innate immune cells are able to bind to the FC portion of the antibody and basically phagocytose whatever the antibody is bound to. And so when it does this, basically it's eating up the virus a lot more efficiently, so you have less virus being able to infect the patient. And again, a lot of this therapy is used in people that are most susceptible, because the main issue with a lot of people that are susceptible is that 5-day delay. If you have a lot of that virus replicating during that time, then you're going to have the immune system basically come in and try to clear it, but there's so much virus that it's trying to clear. There's just going to be a lot of immune cell recruitment and a lot of cytokines being produced, which is going to lead to a lot of tissue destruction, as I highlighted in, I think, the adaptive immunity lecture, where cytokine storm exists. So to prevent that, one of the ways that you're able to treat these infected individuals is by treating them with antibodies that have already been manufactured to recognize the virus instead of your immune system itself doing it. With that, I will take any questions for these different kinds of therapies. It's really important for you guys to understand this, because now you should all be educated on how these actually work, and so you can go ahead and answer some of these questions if any of your family or friends have questions about these. With that, you're free to go. I've included the sign-in sheet in there, and yep.